We use muscles every day to do activities. This woman uses muscles to breathe, that kid in the back that's walking, or to do a cartwheel. Nailed it. The action potential travels down the axon terminal into the synaptic knob. This depolarization opens calcium channels, allowing calcium into the cell. This triggers the synaptic vesicles to fuse with the cell membrane,
releasing acetylcholine into the synapse.
Acetylcholine, released from the axon terminal, binds to the receptors on the sarcolemma.
This action on the receptors of the motor end plate opens channels and releases sodium into the cell.
The influx of sodium cations through the sodium-potassium channel into the muscle fiber causes a depolarization, resulting in the formation of an action potential that spreads across the sarcolemma. The action potential veers into the sarcolemma and into the T-tubules, which activates the calcium release from the terminal cisternae of the sarcoplasmic reticulum and into the sarcoplasm. The next process occurs at a microscopic level. Within the fascicle lies many myofibril, and within the myofibril contains many myofilaments. Myofilaments are made up of 4 contractile proteins: myosin, actin, tropomyosin, and tropomyosin. Calcium binds to troponin, which pulls the tropomyosin away from the active site on the actin. Meanwhile, ATP feeds energy into myosin, placing it into a high energy state. With the active site now exposed, myosin head extends and fuses with the active site, forming a cross bridge. The myosin head flexes to a low energy state, resulting in a movement called the power stroke. This process is repeated as long as sufficient calcium is present. The ATP molecule is broken down into an ADP molecule and a free phosphate. ATP causes the myosin head to break free from the active site. The ATP then converts into ADP, while myosin head becomes energized. Calcium is redeposited into the sarcoplasmic reticulum, attaching to the calsequestrin, covering the active site and relaxing the muscle. Shown here is the sarcomere, the microscopic basic function unit of a muscle that begins at one Z-disc and ends at another. The I band here demonstrates the light band that only contains thin filaments. The A band is a dark band because it contains thick and thin filaments. The H zone contains the M line and the thick filaments. While the acetylcholine is picked up by receptors on the motor end plate, acetylcholine esterase breaks down acetylcholine and redeposits it through the axon and back into the synaptic vesicles.